Hi, my name is Min Xu. I'm going to talk about our work on structural pattern mining in cryo-electron tomograms. As we know, the cell is the basic structural and functional unit of all living organisms. So if we look at the cytoplasm of a cell, we can see that it contains many different macromolecular complexes of different structures and with different spatial organizations. So these macromolecular complexes are nanomachines that governs the wide range of cellular processes. So if we want to accurately know such cell processes, we need to know both the structures and the spatial organizations of these macromolecular complexes inside single cells. Currently, although we are able to obtain the atomic models of a number of macromolecular structures, however, they are not obtained from single cells. And if we think about carefully about how such structures are obtained, we can see there are some major problems. So these structures are often obtained through such popular methods like X-ray crystallography or single particle cryo-EM. And the input of such techniques are the many macromolecules with identical structures. And these macromolecules are obtained through a purification process. And the input of this purification is the cell extract obtained by breaking many cells. As the result of such complicated process, many macromolecular structures are either destroyed or heavily distorted. Therefore, this atomic model may not represent the native structure inside the cell. And also, the spatial localization information of this macromolecule are lost. In general, it has been very difficult to obtain the native structure and spatial organization of large macromolecular complexes inside single cells due to the limits of the data acquisition techniques. However, the recent revolution of cryo-electron tomography technique has made obtain such information possible. So this is an illustration on how this imaging technique works. So first, the samples are frozen and put into a transmission electron microscopy, and we have this electron beams to pass through this sample to obtain a projection image of the sample. And then we can rotate this sample to obtain a series of projection images, and these projection images are used to construct a 3D image of the cell sample at its close to native state and with submolecular resolution. Therefore, this 3D image, this reconstructed 3D image contains both macromolecular structure and spatial organization and their interaction with other cell organelles. And this method become the very promising technique to study the structural biology in situ. We have been working on developing different methods to systematically detect macromolecular structures in such cellular tomograms. And in other words, we have been developing methods for structure pattern mining or in silico purification to do a template-free detection of these macromolecular structures in such image data. Although such Cryo-ET technique shows a great promises for such a systematic detection work, however, there are a number of major limits. So first, there are significant degree of imaging limits, like missing data and low signal noise ratio in such images. And also, because this 3D image technique captures all the macromolecular structures together with ultrastructures and other organelles at once inside a subregion of a cell, there is a great degree of structural complexities. For example, the macromolecular structures are very diverse, and there could be a high molecular crowding level. And also, most of the native structures are known. Thirdly, there is a big data problem. So nowadays, with the automation of the data acquisition, it's not difficult to obtain hundreds of tomograms within a week. And these tomograms can contain millions of macromolecules. And such automation is also quickly improving. So it's therefore we can have a fast accumulation of such image data. So how to deal with this big image data is also pose a very high computational challenge. This is an example of such structural pattern mining analysis results. So these are two slices of two 3D tomograms of two bacterial cells. So by applying our structural pattern mining, we are able to detect tons of different structure patterns, and among which this pattern we found is very interesting. It has a structure that very well fits the crystal structure of this Cryo-EL complex. On the other hand, this pattern fits also very well the low resolution structure of ribosome. In addition, the patterns detected in these single cells also have very distinct spatial distributions. Now I'm going to show you a number of methods that we recently published to support the structural pattern mining as examples. Okay, so one major category of such methods deal with the subtomogram classification. A subtomogram is a subvolume cropped from this big 3D tomogram, and a subtomogram is likely contains one macromolecule. And the subtomogram classification basically is a supervised or unsupervised classification process to determine what class of the structure this subtomogram contains. Previously, we have developed a number of geometric methods for such subtomogram classification. However, these methods are either inaccurate or too slow to deal with the big data problem that exists from the tomogram data acquired using a state-of-art microscope. Therefore, it is very important to develop some methods to efficiently and accurately separate or filter these millions of macromolecules into structural homogeneous subsets. And to do, we first published two subtomogram classification neural network models on ISMB, which is the top computational biology conference. And then we further published three improved models on a journal called Machine Vision and Applications. We then developed a number of methods to deal with different aspects of this subtomogram classification. For example, we have developed our model compression technique to significantly further speed up the classification speed. We have developed a cross-domain adaptation methods so that we can train a supervised subtomogram classifier on simulated data and then adapt it so that it can perform the prediction on real data. This is achieved through this adversarial domain adaptation. And this work was also published on ISMB last year. In order to inspect what are the regions of interest to contribute the classification decision for this neural network model, we have also developed a visualization technique. And this work has been published on MICAI conference, which is the top conference in the biomedical image analysis. Supervised deep learning normally requires large amount of training data with properly labeled structure classes. However, for the cellular tomograms, such data is normally very difficult to obtain. Therefore, it's very important to reduce the amount of annotation needed for such supervised learning. We have developed a semi-supervised learning method that makes use of the autoencoder to include a certain amount of self-supervision to help to improve the classification accuracy and in the same time significantly reduce the amount of subtomogram annotation work. This work was published in BMVC, which is a major, prestigious conference in the computer vision domain. As mentioned before, most of the native macromolecular structures are unknown. Therefore, it's difficult to include such unknown structure classes into the training data for supervised training. Therefore, we have developed an open set learning methods to determine whether two test subtomograms belongs to the same structure class or not, regardless whether the structure class exists in the training data or not. So this work has been accepted as a spotlight presentation at BMVC, which has an acceptance rate of less than 6%. Furthermore, to calculate the statistical confidence on whether two macromolecular structures are identical, we have developed a generative adversarial network-based approach to generate a distribution of randomly sampled pseudo-macromolecular structures. This work was also published on BMVC. The supervised classification of subtomograms requires the availability of a training data set with the subtomograms annotated with their structural classes. However, such annotation are often may not easy to obtain. Therefore, we have also looked at the unsupervised deep learning methods to classify such subtomograms through clustering. So we have developed a method that is based on the convolutional autoencoder. So basically, the convolutional autoencoder has two components, encoder and decoder. So given an input image, the encoder compresses this input image into a low-dimensional vector, and then the decoder decodes this vector into an image. And the autoencoder is trained so that the decoded image is as close to the original input image as possible. And we have constructed a 3D autoencoder and use it to learn this low-dimensional representation of the input subtomograms. And then in this low-dimensional space, we perform clustering to group these input subtomograms into different structural classes. And this work was published on Journal of Structural Biology, which is a popular journal in the structural biology domain. And this work was done by my PhD student Xiangrui Zeng, and he published this paper at the end of the first semester of his PhD study. This is an example on the classified structural patterns embedded to the original tomogram, and we can see that a number of membrane patterns and also blob-like ribosome similar patterns are detected with distinct spatial distribution. Another type of work we have been doing is about the segmentation of subtomograms or tomograms. As mentioned before, the macromolecules could be inside a very crowded molecular environment. And in such case, if we cut out a subtomogram around this macromolecule, then this subtomogram may not only contain the macromolecule of interest, but also some neighbor structures. And the existence of such neighbor structures may bias the recovery and classification of this macromolecular structure of interest. Therefore, we need to segment out the region occupied by this macromolecular of interest from this subtomogram. And previously, we have developed a number of subtomogram segmentation methods based on geometric heuristics, and which was published on ISMB. And recently, we have developed also supervised deep learning methods to perform such classification, which achieve the improved classification performance. The nice thing for deep learning is it's very flexible, and we can construct a multitask learning model to simultaneously perform the segmentation and structure classification and coarse structure recovery work. Okay, and this work has been published also on BMVC. The tomograms contains not only macromolecules but also different cellular organelles, such like mitochondria. Therefore, to study the interaction of macromolecules with these organelles, it's also important to automatically localize such organelles in the tomograms. So to do so, we have also adapted object detection methods, and which is based on the fast RCNN model to automatically localize this mitochondria in these tomograms. In addition, we have developed the Saliency detection methods to identify the regions that stand out from background, so that we can significantly simplify the segmentation task by only focusing on such region. Another type of very important work we have been doing is on this subtomogram alignment. So given two subtomograms which are basically 3D images, so the alignment of subtomograms is to find the optimal rotation and translation of one subtomogram, so that the structures inside these two images can achieve maximum overlap. We have also published at ISMB a method for integrating our previously invented fast subtomogram alignment method and a statistical model for maximum likelihood estimation of the underlying macromolecular structures through subtomogram averaging. So, and this method was published on ISMB. So, and this method essentially uses our previously invented fast alignment methods to populate a number of suboptimal rigid transformations and use it to approximate an integration operation for such a maximum likelihood estimation. Okay, which is the full integration was not computationally feasible, but with such approximation, we can achieve the speed, significant speed up compared to the existing maximum likelihood estimation method, and also achieve a significant quality improvement compared to our previously developed fast alignment method. So in summary, we have been working on developing different computer vision and machine learning methods to facilitate the understanding and analysis of cellular tomograms, which is fundamentally important to understand the basic molecular machinery of cells. For more information, you are welcome to contact me or look at our lab's webpage.